The moment that you realize something that you've worked on since it was in the lab and coming into the clinic helps a patient is like a dream come true. Drug development really requires scientific rigor, discipline. You have to have the expertise. You need to be creative, but there's serendipity, and you need to be ready and to see it. And then that's where like really incredible breakthroughs happen.
The story really began 2 decades ago when BMS was one of the first companies to begin the study of immunomodulatory drugs in blood cancers. They've become the standard of care and continue to have a profound impact for patients' lives. At the time, we had very little insight into exactly how these molecules were working.
Back in 2010, scientists in Japan discovered that immunomodulatory drugs, or IMiD drugs, bound a protein called cereblon, and cereblon is part of the protein degradation system in cells.
Now, the story got really interesting because around 2014, a number of really important publications came out from the Harvard scientists, internal research at BMS, and also our Japanese colleagues, in which we began to understand that not only were these compounds binding to cereblon, but in fact, they were actual molecular glues. It was a light bulb moment. The impact of understanding how the IMiD compound works, it gave rise to a whole new field of targeted protein degradation. And really there was an early understanding that targeted protein degradation, as represented by the IMiD compounds, might in fact represent essentially the tip of the iceberg for what that new modality could bring to patients. At the time, when you see like just an incredible, beautiful piece of scientific research, drug discovery, and clinical development, it's something that it's quite awe-inspiring. As we begin to understand what are sort of the rules of the road for targeted protein degradation, it allows you to begin to think about the medical problem you want to solve, the disease-causing protein that's affiliated with it, and now perhaps intentionally design a molecule, the chemical structure required to create that completely novel molecular glue to recruit that disease-causing protein.
Proteins are the functional molecules in a cell, and the cell needs to regulate their production, modify them, and then get rid of them when they're not supposed to be around. And the getting rid of them is through protein degradation. It's the cell's recycling system. We have several approaches to protein degradation at Bristol Myers Squibb. One of those, and the most advanced, are the CellMod agents. These drugs act as small molecular glues. They bring disease-causing target proteins to cereblon and then target them for degradation. They are the furthest along in our pipeline and nearest term impact for patients. Another approach we have to protein degradation are ligand-directed degraders. Ligand-directed degraders are 3-part molecules. They have a binder that binds cereblon, a linker, and then a binding end moiety that binds the target protein. LDDs have opened up a new target space as well, and it's exciting that we have one that's advanced in solid tumors and reached proof of concept. There's also some disease-causing targets where we can use these degrader antibody conjugates. The antibodies bring the degrader to the specific cell expressing a protein, and only those cells you get the degradation. So it allows a selectivity that can really widen the therapeutic index of those drugs. Having these multiple approaches to protein degradation really allows us to match the right modality to the disease. As we advance these degraders, we're really leveraging our expertise at Bristol Myers Squibb, and that includes using artificial intelligence and machine learning. 100% of these small molecule degraders and LDDs are now being supported by what we call Predict First, where the chemists are being assisted by these machine learning algorithms to predict first which molecules to make and test, and then to feed the algorithm to optimize our drugs to get them to patients faster.
New modalities do not come along very often. And so when they do, there's great promise. And there's also a phase in which the field is going through this incredible sort of rapid and creative expansion. And it's one of the main motivations for our BMS scientists to drive forth novel medications for patients who need them most.
I was inspired to become a physician scientist because I loved science, but also because I had a friend when I was a child who had leukemia. To see him go through that and really the medicines to fail and not help him inspired me to apply what I do to develop new medicines for patients who don't have available therapies. It's been such an incredibly fulfilling journey to see therapies that we've worked on from the earliest days to actually help patients. It just keeps you motivated to keep going, keep working on the next innovation for the patients.